Okay, so the last lesson, Ms. Lum talked about basic Punnett square problems. If you got to this last question and it was about crossing an F2, F2s are when you take any of the babies from the cross above and you mate them with each other. You still have to do all 5 steps, so you would still say the key, who are the parents, whichever 2 babies you pick, those are your new parents, Punnett square, genotype, phenotype ratios. Today, we're going to move on and we're going to start talking about dihybrid crosses. Di means 2. So these are 2 trait crosses where you're looking at, I don't want to just know, is my child going to have blue eyes? I want to know, are they going to have brown hair and blue eyes? So I'm looking at 2 things at the same time. When we do this, there are some laws that are part of dihybrid crossing, and one is the law of probability, the likelihood or chance that something is going to happen. Mendel, the father of genetics, he was an Austrian monk. He studied pea plants. Remember that Ms. Lum talked about the pea plants and the simple fact that there are traits that you may or may not know, and if you can't find them, go back to that coloring, and in the coloring you should be able to identify, is the trait dominant or recessive. Oh, that's okay. So he came up with something known as the concept of alleles. So these are genes on a chromosome, and these genes that are on the chromosome are at specific locations known as loci or locus. We talked about this a little bit. This comes back to the Human Genome Project, where we're looking at different traits in people's DNA. So this is very relevant to what's going on in the world today. Anyway, back to our alleles and Mendel. So we're starting with pea plants, and we're looking at the fact that he started to look at different generations of individuals. He referred to these as first and second filial generations. So this is on that first page of notes that we started with genetics. And it's basically just the first babies or first offspring and the second set of offspring from those generations. There are 2 laws associated with dihybrid crosses and genetics. One is known as the law of segregation. Genes segregate independently and do not influence each other's inheritance. So the seed shape is not indicative because of the pod color. They can be totally independent of each other. The independent assortment is when genes from different traits can segregate independently during gamete formation. So this is the making of those sex cells. If you remember sister chromatids separating, not all of the female sister chromatids went one way and the males the other way. They mixed themselves up, so they ended up creating genetic variation or genetic variability. So we're going to do an example, and we're going to do it in 2 different methods in a moment. Ms. Lum is going to show you the first 2 4-box method with probability and statistics, and I'm going to show you a 16-box method. So we'll look at this problem in a moment. We're looking at 2 separate traits. We're crossing round and wrinkled and yellow and green. So one is what's the texture? The other is what is the actual color. When you flip over to the back, it shows you probability, and this will be the 2-box method that Ms. Lum will show you in a moment. And basically, it's just telling us that the probability that things land on heads or tails, if that's how we're flipping coins, is 1/2 times 1/2 times 1/2 gives you 1/8 of a chance that it will happen. And each event will be independent, so there's a 50-50 chance every time you do this. So Ms. Lum is going to pick up here, and she will show you how to do the 2-box method and then put it together statistically.